Electrical potentials exist across the membranes of all cells of the body. Nerve and muscle cells are capable of rapidly changing membrane potentials, and these rapidly changing potentials, they are used to transmit signals along the nerve or the muscle membranes. You should also know the concept of diffusion potentials. A concentration difference of ions across a selectively permeable membrane can create a diffusion potential. As you can see here in the diagram, that all cells of our body, they have a sodium potassium pump, and this sodium potassium pump, it transfers potassium ion from outside to inside, while sodium from inside to outside. So naturally, potassium ion, it has a tendency to move from the inside to the outside whenever it is given a chance, and the sodium ion, it has a tendency to move from outside to inside, because the sodium potassium pump, it has created a concentration difference across the membrane of these ions, that sodium is always in greater amount outside the cell, so it always has a tendency to move from outside to inside. And when the channels, the voltage gated channels, they are opened, and in any other way sodium ion gets a chance to move from one side of the membrane to the other, then it moves from the outside to the inside till the point the inside of the membrane, it becomes positively charged. And in case of sodium, when it becomes around plus 61 millivolts, only then this transport of sodium ion from the outside to the inside will stop. Otherwise, the sodium ion, it will keep trying to diffuse from the outside to the inside. And now the similar case of the diffusion potential which occurs with the potassium ion, it is that because of the working of the sodium potassium pump, now concentration gradient, it has been created where the concentration of potassium is much more inside as compared to that on the outside. So potassium, it has a diffusion potential, a hidden energy to diffuse from the inside to the outside whenever it gets a chance. So, what is Nernst potential? The diffusion potential level across a membrane that exactly opposes the net diffusion of a particular ion through the membrane, it is called Nernst potential. And here you can see the formula for calculating the Nernst potential of an individual ion. It is assumed that the potential in the extracellular fluid outside the membrane is at zero potential, and the Nernst potential is the potential inside the membrane. Now, the sign of the potential, it becomes positive if the ion diffusing from the inside to the outside, it is a negative ion. And similarly, the sign, it is negative if the ion diffusing from the inside to the outside, it is a positive ion. The Goldman equation is used for the calculation of the diffusion potential when the membrane is permeable to several different ions. When a membrane is permeable to several different ions, the diffusion potential that develops that depends on three factors: the polarity of the electrical charge of each ion, then there comes the permeability of the membrane to each ion, which is represented by this capital P alphabet, and also the concentrations of the respective ions on the inside and also on the outside of the membrane. This Goldman equation, or the Goldman-Hodgkin-Katz equation, it gives the calculated membrane potential on the inside of the membrane when two univalent positive ions, sodium and potassium, and one univalent negative ion, chloride, they are involved. Sodium, potassium, and chloride ions, they are the most important ions involved in the development of membrane potentials in nerve and muscle fibers and in the neuronal cells in the nervous system. The concentration gradient of each of these ions across the membrane, it helps determine the voltage of the membrane potential. The degree of importance of each of the ions in determining the voltage, it is proportional to the membrane permeability for that particular ion. If the membrane it has zero permeability to both potassium and chloride ions, the membrane potential is determined by the concentration gradient of sodium ions alone, and the resulting potential it will be equal to the Nernst potential for sodium. A positive ion concentration gradient from inside the membrane to the outside, it causes electronegativity inside the membrane. The reason for this is that the excess positive ions diffuse to the outside when their concentration is higher inside than outside. This carries positive charges to the outside, but it leaves the non-diffusible negative anions on the inside, creating electronegativity on the inside. The opposite effect occurs when there is a gradient for a negative ion. A chloride ion gradient from the outside to the inside, it causes negativity inside the cell. This is because the excess negatively charged chloride ions, they diffuse to the inside, leaving the non-diffusible positive ions on the outside. The permeability of sodium and potassium channels, it undergoes rapid changes during the transmission of a nerve impulse. The permeability of the chloride channels, it does not change greatly during this process. Therefore, the rapid changes in sodium and potassium permeability, they are primarily responsible for the signal transmission in neurons. Right, now you are looking at a university question. The question will be something like define the resting membrane potential and how it is generated. Or it could be something like explain the mechanism for the development of resting membrane potential. The resting membrane potential of large nerve fiber at rest is about minus 90 millivolts. It means that the potential inside the fiber is 90 millivolts more negative than the potential in the extracellular fluid on the outside of the fiber. As far as the cause of the resting membrane potential is concerned, you should know that resting membrane potential is due to the diffusion potential caused by the diffusion of diffusible ions such as sodium and potassium across the membrane. Because of their unequal distribution across the membrane, means whether it is inside or outside. This unequal distribution, it occurs due to the presence of negatively charged anions such as proteins, organic phosphate and sulfate inside the cells. As the cell membrane, it is not permeable to these anions. All the cell membranes of our body, they have a powerful sodium potassium pump that continually transports sodium ions to the outside of the cell and potassium ions to the inside. The pump has a vital role in cellular survival because it prevents cell swelling by moving 3 sodium out of the cell for every 2 potassium pumped into the interior. Um, Now, how does it play a vital role? You see, a large number of the proteins and other organic molecules, they are present inside the cell, and these cannot escape from the cell because of their large size. And further, these molecules, they are mostly negatively charged. So negativity, it attracts large number of potassium, sodium and other positive ions. Now all these molecules and ions, then they cause the osmosis of water to the interior of the cell. So when the sodium potassium pump, it transports sodium out, which due to the less permeability, it stays out. So what happens that when more sodium it gets out, it causes a net loss of ions out of the cell. This loss, it initiates the osmosis of water out of the cell, and this is how the cell swelling is prevented. One more interesting bit you should know is that the sodium potassium pump, as I might have already told you, this is an electrogenic pump because more positive charges, they are pumped to the outside than to the inside. The sodium potassium pump causes large concentration gradients for sodium and potassium across the resting nerve membranes, as you can see in this figure. Sodium is concentrated outside the membrane, while potassium is in large quantity inside the membrane. There is a channel protein called a potassium leak channel in the nerve membrane through which potassium can leak even in a resting cell. These potassium leak channels, They are much more permeable to the potassium than to the sodium, normally about 100 times more permeable. This difference in permeability, it is a key factor in determining the normal resting membrane potential. Now, in the origin of resting membrane potential, certain important factors, they are contribution of potassium diffusion potential, sodium diffusion potential, and the contribution by sodium potassium pump. Keep in mind that potassium has a major role in the genesis of resting membrane potential. The nernst potential for potassium is closer to the resting membrane potential of minus 90 millivolt in nerve fiber. And it provides proof that potassium is the main ion responsible for resting membrane potential. Nernst potential for sodium is far apart from the resting membrane potential, so sodium is not main ion which is responsible for maintaining RMP. Now, we will look at the contribution of potassium's diffusion potential. We assume that the only movement of ions through the membrane is the diffusion of potassium ions. And you can see that potassium is concentrated on the inside as compared to that on the outside. So it has a diffusion potential to transport from the inside to the outside of cell. The ratio of the potassium ions from inside to outside the membrane, it is around 35, and this gives a calculated Nernst potential for the inside of the membrane of minus 94 millivolts, as you can see in this equation. We took the ratio of the potassium ion concentration inside to outside, and then this ratio, the log for this ratio was taken and it was multiplied with 61, and we got the Nernst potential for the potassium, which is minus 94 millivolts. You see the logarithm of 35, it is 1.54, and this is multiplied by 61, and then it becomes minus 94 millivolts. Therefore, if potassium ions, they were the only factor causing the resting potential, the resting potential inside the fiber, it would be equal to minus 94 millivolts. Do you wonder what could be the contribution of sodium's diffusion potential? Now, this figure shows the permeability of nerve membrane to sodium ions caused by the minute diffusion of sodium ions through the potassium sodium leak channels. The ratio of sodium ions from inside to outside the membrane, that is 0.1. which gives a calculated nernst potential for the inside of the membrane of positive 61 millivolts. Applying the Goldman equation gives a combined potential inside the membrane of minus 86 millivolts. The sodium potassium pump, it has its own contribution in the dynamics of resting membrane potential. You all know that sodium potassium pump, it moves more sodium ions to the outside than the potassium to the inside. So this causes an additional degree of negativity of minus 4 millivolts inside the cell. Let's now calculate the resting membrane potential keeping all the contributions in mind. You see the potential which is contributed by the diffusion of potassium ions alone, that is around minus 94 millivolts. The potential which is contributed by the diffusion of sodium ions alone, that is positive 61 millivolts. While the potential which is contributed by the diffusion of both potassium and sodium ions, this is minus 86 millivolts. Now, the potential which is contributed by sodium potassium pump, it is minus 4 millivolts. So, the resting membrane potential of the nerve fiber, that is minus 86 and minus 4 added up, which makes it minus 90 millivolts. So now you see, my dears, that how a resting membrane potential is developed. Let's now start with the nerve action potential, for which you might get a question like the one shown on screen in your university examination. Nerve signals, they are transmitted by action potentials, which are rapid changes in the resting membrane potential. Each action potential begins with a sudden change from the normal resting negative membrane potential to a positive potential. The action potential ends with the equally rapidly back to the negative potential. Now, let's look at the stages or phases of the action potential, which are resting, depolarization, and repolarization stage. Uh, this is the resting membrane potential before the action potential begins. And in this resting stage, the membrane is said to be polarized. In depolarization stage, the membrane suddenly becomes permeable to positive ions, such as the sodium ions. Large number of positively charged sodium ions, they diffuse to the interior of the axon, or you can say the interior of the cell. The potential rises rapidly in the positive direction. The change in the voltage, it stimulates the opening of additional voltage gated sodium channels. Now here you can see this term overshoot. The membrane potential, it may overshoot beyond the zero level and to become somewhat positive, because as the additional sodium it rushes in, the membrane potential actually reverses its polarity so that the outside of the membrane is negative relative to the inside. Now, this depolarization stage, it may also be labeled as a rising phase of action potential. How does the repolarization stage starts, which is also called a falling phase? You see, within a few 10,000ths of a second, the sodium channels begin to close and the potassium channels open. Diffusion of potassium ions to the exterior re-establishes the normal negative resting membrane potential. Here, I wouldn't miss an important concept of after hyperpolarization or undershoot. Since many more potassium channels have been opened than are required and not all close when the membrane potential returns to normal resting level, and that causes an undershoot. So, this hyperpolarization persists until the membrane permeability to the potassium it returns to normal. Voltage gated channels have a vital role in action potential, so let's dive in their detail. Voltage gated channels are of two types: the voltage gated sodium channels and the voltage gated potassium channels. Now keep in mind that these voltage gated channels, they are in addition to the sodium potassium pump and the potassium leak channels. Voltage gated sodium channel, it has two gates, one outside the channel, and this is called activation gate, while another inside the channel, and this is called inactivation gate. At the resting membrane potential of minus 90 millivolts, the activation gate is closed. So, what causes the activation of sodium channels? In that regard, a threshold stimulus causes the voltage to rise between minus 70 and minus 50 millivolts. And this causes a sudden conformational change in the activation gate and opens it. And this is called the activation of sodium channels. Now don't forget that the sodium channels, they have to be inactivated also, and this phenomenon is time controlled. This inactivation gate, it closes a few 10,000ths of a second after the activation gate opens. After the inactivation gate closes, sodium ions cannot get inside. So the membrane potential, it begins to recover back towards the resting membrane state. Keep in mind that this inactivation gate, it will not reopen until the membrane potential returns to or it is near to the original resting membrane potential level. Let's now move on to voltage gated potassium channels. This channel has one gate only, which is present on the inside of the channel. At the resting membrane potential of minus 90 millivolts, the gate is closed. For the activation of voltage gated potassium channel, when the membrane potential it rises from minus 90 millivolts towards zero, it causes the opening of gate and it allows increased potassium diffusion outward through the channel. The decrease in the sodium entry to the cell and the simultaneous increase in the potassium exit from the cell, it combines to speed up the repolarization process. So this is how the repolarization of the membrane is achieved.